Hello friends, in our previous lectures we learned about the classification of subdivision Deuteromycotina and very important characteristics of the class Hyphomycetes. In today's lecture we are going to learn about the important characteristics of the class Coelomycetes. According to Ainsworth classification, the class Coelomycetes is further divided into two form orders, that is Spheropsidiales and Melanchoniales. The Spheropsidiales is further divided into four form families, that is Spheropsidaceae, Excipulaceae, Nectrodiaceae and Leptostomataceae. Coming to the important characteristics of class Coelomycetes. Here the conidia are born on conidiogenous cells with or without distinct conidiophores, that is enclosed in a fungal fructifications such as pycnidium or acephalus. These are like a fruiting bodies, asexual fruiting bodies. So here conidiophores may or may not be present. And the Coelomycetes is already told divided into two form orders, that is one is Spheropsidiales and second one is Melanchoniales. So coming to the characteristics of the Spheropsidiales, the fruiting body is generally what we call as a pycnidia. So pycnidia looking like it used to be a globose or flask shaped asexual fruiting body which is lined inside with conidiophores. So conidiophores may present, may not be present, but if it is a here you can see it is a pycnidial body. Inside it is lined with the conidiophores on which conidia are produced. So it may be completely closed or may have an opening. Here you can see the opening. This opening is known as ostiole. Sometimes it may be completely closed without having any opening. It may be papillate or beaked or long necked at the apex. So this apex, so this pycnidium may be globose or maybe it may be is a flask shaped structure which is having the beak at the apex, and they vary greatly in their shape, that is shape of the pycnidium and size of the pycnidium, what is the color and consistency of the pseudoparenchymatous wall also. And here this pycnidium is made up of pseudoparenchymatous tissue. So based on this pseudoparenchymatous tissue, the shape, size and color of this pycnidium vary in the different genera. The Spheropsidiales further divided into four form families, that is Spheropsidaceae, Excipulaceae, Nectrodiaceae, which is also known as Dithiaceae, and Leptostomataceae. So let us see what are the different genera included under these different form families. Under Spheropsidaceae, the pycnidia look like a flask shaped or globose, thin or thick walled. It may be with the loose pseudoparenchymatous tissue or it may be thick walled and dark colored osteolate, that is opening is present at the apex and hard texture. The different genera included under the Spheropsidaceae are Phoma, Phomopsis, Macrophomina, Philosticta, Septoria, Diplodia and Botryo-Diplodia. So let us discuss about the individual characteristics of these genera. Coming to the first one, Phoma. Phoma pycnidia look like very small and they are dark colored, generally immersed in host tissue. So this pycnidia used to be globose or flask shaped. Generally pycnidial shape used to be globose or flask shaped in nature, and here flask shaped pycnidia used to be very thin walled and osteolate. Here osteole is present in the Phoma species, and the wall of the pycnidium consisting of a dark pseudoparenchymatous cells. And coming to the conidiophores or conidiogenous cells, they are very short in nature. Already in previous picture what I showed, they are very short in nature and hyaline, lining the inner pycnidial wall and producing the conidia in succession. So continuously they used to produce the conidia. Generally where these conidiophores are laid, they are laid on the inner side wall of the pycnidium. And conidia are hyaline, that is light in color, aseptate, they do not have any septation, and gutulate, that is they are having oil globules in the conidium, and pyriform to globose. So size this shape of the conidia used to be vary from pyriform to globose, and how they will ooze out through the pycnidia means like a long thread like cirrus through the ostiole. Through ostiole these conidia are continuously ooze out like a one continuous stream like structure. So important examples are the Phoma lingam, which used to cause a black leg of crucifers. In this picture you can see the Phoma lingam causing a black leg in case of crucifers. Coming to the Phomopsis, here Phomopsis pycnidium looks brown to black in color, and it is a globose and papillate osteole with one or more locules, and pycnidium may contain one or more locules in case of Phomopsis. Conidiophores or conidiogenous cells are simple or branched. Here we can see the branching of the conidiophore cells, whereas Phoma we could not see the branching, only simple conidiophores. And here conidia are of two types. Here in this first picture you can see alpha conidia and beta conidia. Alpha conidia are hyaline and they are ovoid. Here these are the alpha conidia, they are ovoid in shape and they are one celled, whereas beta conidia they are hyaline and filiform, that is eagle shaped conidia, and straight, either they may be straight or they may be curved, and but they are single celled. Example for the Phomopsis are the Phomopsis vexans, which used to cause fruit rot and blight of brinjal. Here you can see the symptoms on the fruit caused by the Phomopsis vexans. Coming to the third genus, Philosticta. Philosticta pycnidia, the dark osteolate and globose in shape, and it is immersed in the host tissue and erempent or with a short beak. This pycnidia is having the short beak, and conidiophores are generally short and obsolete, and conidia are also small, they are one celled and they are very light in color, and conidia are maybe ovoid in shape to elongate. So ovoid or elongate, they have different shapes. And here important character what we have to remember is Philosticta and Phoma are differentiated on the basis of plant organs attacked. Here Philosticta generally used to affect on the leaves causing, and it used to cause leaf spot diseases, whereas Phoma occurs mainly on stem, twigs and fleshy roots. So Philosticta used to cause leaf spots and short holes on the leaves, whereas Phoma used to occur mainly on stem, twigs and fleshy roots. Example, Philosticta gingivaris, where it used to cause leaf spot of ginger. And here you can see the differences of the pycnidial bodies of the Phoma and Philosticta. Here Phoma is also osteolate, and here Philosticta. Here this upper side you can see how this conidia ooze out from the pycnidial bodies. And coming to the Macrophomina, Macrophomina is the most important pathogen which used to cause common disease charcoal rot in many of the crops. So Macrophomina pycnidia used to be very globose and dark brown, and it also having the papillate osteole, that is it has having the opening. Conidiogenous cells are the barrel shaped and hyaline. Conidia also hyaline aseptate, they are cylindrical to fusiform in shape. Under laboratory conditions, here on the media you can see this Macrophomina used to produce sclerotia. Here on the microscope you can see sclerotial bodies. So example for the Macrophomina is Macrophomina phaseolina, which used to cause charcoal rot, canker, damping of jowar, maize and groundnut. Coming to the next species, that is next genus, that is Septoria. Septoria pycnidia is also immersed in the host tissue, and this pycnidia is globose, brown in color. Here the pycnidia is a thick walled, papillate and osteolate. Most of the genera is having the common characteristics, only the conidial shape it may vary, and this pycnidial lining, whether it is thick shaped or it is a thin in size, like that it will vary. And conidiophore or conidiogenous cells are hyaline, broad and round. Here conidiophore, you remember that it is a broad and round at the base and narrow above or barrel shaped, that is at the base it used to be round shape, at the top it used to be barrel shape, that is narrow. And conidia are here Septoria, conidia are septate. Before that what we saw in Phoma, Philosticta, these are aseptate conidia, but here in Septoria, the name itself indicating Septoria, that septa is present in the conidia, and conidia are filiform. Here in this picture you can see septation in the conidia and filiform, that is a sickle shaped type conidia we can see here. Examples are the Septoria nodorum, which used to cause gloom blotch of wheat, and Septoria lycopersicea used to cause leaf spot of tomato. And coming to the another genus, Diplodia. In Diplodia, pycnidium used to be black, globose, papillate and osteolate. Here you can see And conidiophore or conidiogenous cells are slender, they are hyaline and cylindrical, and here important characteristics is conidia, which is used to be brown and two-celled here, and ovoid here, and here you can see round at the tip. So obtuse, round, and base truncate, that is here you can see a round shape is there and it is a two-celled. And example for this diplodia is Diplodia natalensis, which used to cause Diplodia gummosis of the citrus. Coming to the next genus Diplodia, where pycnidium is carbonaceous and dark brown or black with no osteole. This is the important character in case of Diplodia, whereas Diplodia osteolate is there, whereas in Diplodia pycnidium is devoid of the osteole. Conidiophores are simple and very short in nature. Here you can see Diplodia and conidia are dark brown, two-celled and ovoid. Here you can see the conidia which is two-celled and ovoid in shape, and they are dark brown in color. And here they are like looking like locules, Diplodia pycnidium, which is containing locules. Example are the Diplodia theobrome. This is very important disease in case of sapota, which used to cause flat limb of sapota. So important characteristics of the family Excepulaceae. So we already covered one family, now another family Excepulaceae. In Excepulaceae pycnidia are cup shaped. In previous class it is globose in shape, here it is a cup shaped pycnidia. Example are the Aphelis, where Aphelis pycnidium is a cup shaped, and conidia are hyaline, one celled, and acellular, that is needle shaped conidia. Example are the Aphelis oryzae, which used to cause Udabatta disease of rice. And coming to the another family Nectroidaceae, here pycnidia resembles the perithecia of the Nectria, and hence the family name they gave it as a Nectroidaceae. Example Gythia. In Gythia, where pycnidium is a flask shaped and it is a colored and it is a soft texture, that is it is fleshy in nature, and osteole is already present for this pycnidium. And conidia are hyaline, aseptate, oblong and rounded at each end. Only septoria is having the septate conidia. And example for the Gythia fragaria, which used to cause leaf blotch and stem and rot of strawberry. Here you can see the symptoms of this Gythia. Coming to the another family Leptostomataceae, the name itself indicating that stomata formation used to be there. And pycnidia are shield shaped or elongated or flattened. So this pycnidia is either shield shaped pycnidia, or it may be elongated, or it may be flat. And genus is Leptotherium. So same pycnidia, that is shield shaped, dark, and one half smaller than the other. Here you can see one half is smaller, one half is larger. And conidiophores are simple. Conidia, here you can see septate and falcate. It is like looking like a sickle shape, and here it is one celled, but it is a septate. Example are the Leptotherium pomi, that is a fly speck of the apple. So these are the characteristics of the class Coelomycetes. Thank you for your keen interest in this video, and remember to like and subscribe for more videos. If you feel that this video is more important, please share with your friends. I wish you great success, health, prosperity and happiness. Thank you.